My name is Jim Beran. I'm an independent researcher who lives in Montara, California, working on biological evolution of consciousness. My talk is about hypotheses that include protein interactions. In the picture, I'm the guy on the right. Also in the picture are my grandsons and one of their pets. My thanks to those listed on this slide for their help. And last but not least, my collaborator, Joni Diarc, an above-average Norwegian rat of French descent. What makes Joni special and above average? Not only is she a master chef by day, but at night she works as a neuroscientist studying biological evolution of consciousness. We'll talk more in a minute about her anti-ratatouille conjecture, abbreviated ARC. So what is the milieu around Chez Joni? As you can see, Joni's restaurant is in a little-known, almost virtual French Quarter of Tucson. Chez Joni specializes in creations that reflect actual evolutionary paths to consciousness. Joni's not the only one in this field. Other researchers have previously made a number of proposals, some of which are summarized here. But Joni had an insight that might be new. Joni's flash of insight was that the various proposals are not all inconsistent. Multiple types of proteins could play roles in evolution of consciousness. Did multiple types of proteins play a role in evolution of consciousness? Intuitively, it seems the answer should be yes. And that answer is supported by the recently published omnigenic model of evolution. Based on detailed analysis, researchers have found that huge numbers of DNA variants contribute to inheritance of complex traits. And consciousness might be the most complex trait of all. Many of the variants occur in genes, the parts of DNA that are expressed as proteins, but even more variants occur in non-coding DNA, such as in regions that regulate gene expression. So, what did evolution do with those many genes and their counterpart proteins to obtain conscious animals? Did it toss them into a pot and stir, like making ratatouille? Joni doesn't think so. Gene and protein ratatouille is not on her menu. Joni proposes that DNA and proteins changed so that proteins interact in a new way, causing a group of neurons to operate in a new way, changing conscious experience. Starting with Joni's conjecture, we can articulate a 4-scale summary of evolution of consciousness as shown here. Besides the changes at genome variation scale, subneuron scale, superneuron scale, and brain scale, the changed DNA was preserved in subsequent generations so that evolution occurred. Chez Joni has been able to stay open despite the pandemic, sometimes with takeout only. Its menu has 2 parts. First, you choose a sauce, such as a conserved protein, And then you choose a combination or combo of evolving proteins that interact somehow among themselves and with the conserved protein. This two-step approach is helpful because it avoids the need to consider every possible combination of proteins. First, let's consider the sauces on offer at Chez Joni. These are broad categories of hypotheses about evolution of consciousness. Joni divides them into tubulin-based and non-tubulin-based hypotheses. Non-tubulin-based hypotheses are interesting. We'll briefly talk about one later. But hypotheses based on tubulin are the best we've found so far. That's why Chez Joni's specialties are based on tubulin. This diagram illustrates a tubulin-based structure that is abundant in neurons, the microtubule. And here are some reasons we like tubulin-based hypotheses. Tubulins are stable and central to neuron operation. They have many interactions, and tubulins and microtubules are much studied. Now to the second part of the menu, the protein combos. Chez Joni is currently offering 3, A, B, and C. Protein combo A adds 2 types of proteins to the tubulin sauce, ankyrin and ion channels. At the top of this diagram, a dark blue protein extends through the cell membrane. That is a voltage-gated sodium ion channel. Inside the cell, the orange protein connected to the ion channel is an ankyrin called ankyrin G. And the gray structures connected to the ankyrin G at the bottom are microtubules. Here is our 4-scale tubulin-based hypothesis for combo A. Ankyrin and ion channels evolved features that allow them to bind. Ankyrin can therefore connect bound ion channels to microtubules, an example of a new protein interaction, and can concentrate ion channels. In response to voltage, concentrated ion channels can initiate an action potential and also affect extracellular electrical signals in a neuron group. The neuron group's changed signals in turn change the brain's electrical and/or magnetic pattern, here abbreviated EM. According to this hypothesis, the changed EM pattern in turn affects conscious experience. That sounds like a good start, but it's not the end of Joni's menu. Let's consider protein combo B. Protein combo B appeals to connoisseurs of coupling. I mean EM coupling. This schematic diagram suggests how electrical and/or magnetic coupling could occur between parts of different neurons. A protein structure in the left dendrite would respond to a synaptic signal or to a signal received by coupling from another neuron, and in response would provide a signal that couples to a protein structure in the right dendrite. How might evolution produce electrical and/or magnetic coupling across neurons? Our hypothesis for protein combo B is based not only on tubulin itself, but also on a way in which tubulin is modified, enabling EM coupling. Tubulin itself has not changed much for millions of years, but after tubulin is produced from DNA, it can be modified by attaching or removing amino acids, often in the form of side chains that extend outward from the main chain of amino acids. These modifications are called post-translational modifications, abbreviated PTMs. This schematic diagram shows some of the PTMs of tubulin, and we're especially interested in one of them, glutamylation, which adds an amino acid called glutamate to tubulin. Where a side chain has more than one added glutamate, the modification is sometimes called polyglutamylation. Tubulins in brain tissue are often polyglutamylated. The result is a negatively charged glutamate side chain on the exterior of a microtubule. What controls polyglutamylation? We call proteins that regulate post-translational modifications PTM reg proteins for short. This diagram shows how three types, CSAP, TTLLs, and CCPs, are believed to regulate the amount of glutamylation in a microtubule in some vertebrates, possibly in humans. Polyglutamylation is especially frequent at the high level of glutamylation shown on the right of this diagram. See how the side chains are substantially longer than at the lower levels. Our tubulin-based hypothesis for combo B relies on evolution of PTM reg proteins like these to obtain polyglutamylated microtubules that provide electrical effects. How are such electrical effects possible? This schematic diagram shows one possibility. Polyglutamylated microtubules might provide transient electrical currents due to changes in negatively charged glutamate side chains, referred to in this diagram as tails. When the tails are fully extended due to their mutual repulsion, they might hold ions in place, preventing ion movement along the microtubule. Then, in response to a synapse signal or in response to EM coupling, the tails might become disordered, allowing the ions to flow, providing a transient electrical current shown by the small letter I. Such transient electrical currents in neighboring neurons might couple electrically or magnetically, or both. Our four-scale tubulin-based hypothesis for protein combo B can be summarized as on this slide. A key part is that change in the brain's EM pattern affects the brain's conscious experience. Next, we consider protein combo C, which appeals to those with a taste for complexity. This diagram and the next one reflect the detail and complexity of our hypothesis for protein combo C. Here we see some of the proteins and protein interactions in a neuron's axon initial segment, or AIS. The AIS is often positioned at the beginning of the neuron's axon, close to the neuron's soma or body, as shown in the upper part of the diagram. In this diagram, microtubules are shown in gray at the bottom. Ankyrin G molecules are shown in red, extending from the microtubules to the membrane around the AIS. And connected to the ankyrin G, but extending through the membrane, is a light blue voltage-gated sodium channel labeled NAV. In the large family of bilaterally symmetric animals called bilaterians, giant ankyrin molecules organize the AIS and recruit ion channels and other proteins to it. In humans and similar animals, ankyrin G is the giant ankyrin that does this. The AIS initiates action potentials in the axon, and I have previously proposed that the AIS supports changing conscious experience. This diagram shows examples of additional proteins in protein combo C. End-binding proteins, abbreviated EBs, are examples of microtubule-associated proteins, or MAPs. TRIM46 is one of a family of tripartite motif-containing proteins, and neofacin 186, abbreviated NF186, is an example of an adhesion protein. The end-binding proteins allow one end of ankyrin G to bind to microtubules. Neofacin 186 binds to the other end of the ankyrin and also extends through the neuron's plasma membrane, providing connections to external structures. And TRIM46 mediates so that AIS microtubules form structures called fascicles. After a whirlwind introduction to the AIS, here's an equally whirlwind summary of our four-scale tubulin-based hypothesis for combo C. In this hypothesis, AISs of adjacent neurons are linked and positioned in new ways, enabling better EM coupling between current-like effects in their microtubule fascicles. Better coupling synchronizes action potentials within a neuron group, and synchronization changes the brain's EM pattern. This in turn changes conscious experience. We now have hypotheses for each of the three protein combos on Joni's menu. All three hypotheses are based on tubulin, but they are quite different. Here we apply some familiar criteria for scientific hypotheses. Simplicity favors combo A. Boldness and falsifiability, both recognized by the philosopher Karl Popper, favor combo C. Let's consider one more criterion. Which hypothesis most closely follows the actual evolutionary path? Under this criterion, the best hypothesis is closest to the actual path. It offers the greatest explanatory power. Under the omnigenic model of evolution mentioned earlier, heritability of complex traits is due to a huge number of variants with tiny effect sizes. Because combo C has the most types of proteins and interactions, it seems the most consistent with the omnigenic model. Therefore, it seems the best unless it can be falsified somehow. But a more detailed protein combo could be even better. Now, as I mentioned earlier, occasionally Joni's menu includes a non-tubulin-based hypothesis. Following a suggestion by Professor David Edelman, this hypothesis is based on opsins, proteins that respond to light. Signals at subneuron scale change shape of a lens over the retina. Groups that include retinal cells and sensory motor neurons provide greater acuity, perhaps on an array of neurons. And when acuity becomes sufficient, the brain begins having conscious visual experience. Note the role in this hypothesis of intercellular signaling, which was not present in our previous hypotheses. Here are our conclusions, based mainly on our comparison of hypotheses for protein combos A, B, and C. In short, detailed protein interaction hypotheses can be bolder and more falsifiable, but are generally not simpler. And they can be better if they more closely follow the actual evolutionary path. Here are our thoughts about possible future work. Thank you for your attention. We hope you find an interesting and tasty hypothesis on Joni's menu. And please contact me by email for further discussion, for future access to the video file, or for complete citations.
Hello. Greetings from Greece to all the participants of the conference. I am Paraskevi Papadopoulou, and I am to present Quantum Evolution Caught in Action. Why and how specific adaptations of major plant divisions are similar to Chordata Vertebrata classes adaptations. Here is the outline of the presentation. I am Professor of Biology at DIRI, the American College of Greece. I am also the head of the Science and Mathematics Department and head of the Biomedical Sciences program at DIRI. I have a Bachelor of Science in Biology from UCLA, Ptychion in Biology from the National Kapodistran University of Athens, and a PhD in Biology from National Kapodistran University of Athens. Below you can see my research interests. Here are two representative publications of mine that are related to my current study. The first is on molecular and supramolecular architecture of the Salmon-Gardineri proteinaceous eggshells during development. The second is on laser Raman and infrared spectroscopic studies of protein conformation in the eggshell of the fish Salmon-Gardineri. Here you can see electron micrographs of the eggshell of the trout as it gradually becomes developed, and when it is fully formed is mostly proteinaceous in nature, and the proteins are within lamellae that you can see here quite clearly, and those lamellae contain fibrous proteins. And these fibrous proteins, it's very interesting to see, they are also found in eggshells of species like insects, and in the silk moths especially, it's very obvious that there is a great degree of similarity, and so we have proposed a common helicoidal architecture for all these eggshells. So this is a very important finding from an evolutionary point of view. A number of researchers have worked on these fibrous proteins that come from superfamilies of genes and subfamilies. Shen and his colleagues annotated 127 chorion genes, and these silk moth chorion genes can be seen as one of the best models for understanding the spatial and temporal control of expression of eukaryotic multigene families. Egg envelope proteins serve as a good example of conserved gene origin. And the question that I could ask is whether all this can be explained by Darwinian evolution theory, or could it be guided by quantum evolution as well in a complementary fashion. Overall, I have high interest in studying protein structure and conformation in relation to function, and over the years I have used different methods and techniques like TEM and SEM and biochemical, X-ray diffraction, laser Raman and infrared spectroscopy and molecular techniques. So I am acquainted with the use of interdisciplinary and transdisciplinary approaches. I have also been inspired by the variety of courses that I teach over the years, but for the particular study, the course Cell and Molecular Neurobiology and the course Human Consciousness from Brain to Subjectivity have led to the study that we sent to Barcelona in the Human Brain Project International Conference, Understanding Consciousness, a Scientific Quest for the 21st Century, And the title of our work was The Evolution of Consciousness as a Function of Increasing Neural Complexity. Right after that, the year after, we have sent to the conference The Science of Consciousness in Interlaken, Switzerland, the work titled Can Artificial Intelligence Ever Be Conscious? Now, consciousness is a complicated phenomenon, and it is obvious that the physicists have contributed a lot with their standard model, where we begin to understand quite well what are fermions, what are bosons. We understand the three forces of nature, how and why they are united, and why gravity is still outside this model. According to string theory, perhaps supersymmetry is the model that acts like an expansion pack to the standard model and could help explain some of its unanswered questions, such as why particles have mass. But also, there is a very interesting theory in physics, the quantum field theory, that tells us that there are really no particles at all, only fields, and that particles are merely waves in that field. There is also a very interesting YouTube video, What is the Universe Really Made Of? Quantum Field Theory Visualized, and it's a very interesting, I believe, video to see. But we have to keep in mind that fields may be just an approximation of a deeper level of reality. And this brings us to David Bohm and what he proposes as quantum potential. According to him, the universe and everything, and our perception of the so-called vacuum state, is where particles interact with, respond to, and are informed by information potential, which allows the cosmos to emerge. So it is the information informing action of the quantum potential that makes it possible for the physical universe to be everything we know and everything we will come to know, and that is already information, waiting to unfold into manifest reality. In other words, it is what Bohm says, the implicit waiting to become explicit. Again, there is a beautiful film, Infinite Potential, The Life and Ideas of David Bohm, that is worth seeing. Interestingly enough, information in English means to give form to the mind, but in Greek, information is more than that. It literally means bears fully or conveys fully. And in addition to that, one can say that we could have other theories like the orchestrated objective reduction theory, and from that video, anesthesia, consciousness, Bohm and Penrose theories and Stuart Hameroff's theories are compared. For either, though, the brain must sustain quantum coherent superpositions long enough for cognitive functions. And so could the answer to the mystery of consciousness be related to the infinite potential or even the quantum gravity of the orchestrated model? So we definitely have come up with a number of theories, approaches, if you want, to understanding mind and consciousness. I would say most of them fall within these three major categories as materialistic monism, dualism, or monistic idealism. In the first case, consciousness emerges out of matter, and there is really no free will, and consciousness may be an illusion as well under materialistic monism. But there are other views, and there were poets and philosophers and theologians and mystics that have always insisted that this game of the universe has both players and purpose. So there is this hope that one day scientists might discover a fundamental law of the universe that would not wholly be mechanical, but in some sense free. So there is dualism, matter plus mind, but classical physics actually was forced to accept materialistic monism because of that, because of the dualism that is behind it, and also in this case we have secondary phenomena like epiphenomena that appear. In the monistic idealism is the perspective that is examined in this study. In other words, that mind or consciousness is primary and matter energy arises out of mind. And that means the physical world is to the greater mind as a dream image is to the individual mind. For sure, I had to examine a good number of theories of consciousness, and they are listed here as groups. There is a link that takes us to a nice paper, a review paper, which covers most of them. But there is also this other paper that brings forward the need to establish hard criteria for empirical theories of consciousness, so we narrow down the list. So, this study is the result of a thorough literature review of the present-day theories of consciousness as they might apply to morphogenesis, the natural selection evolutionary process, convergence and coevolution. It's important to remember when talking about evolution that we live in a universe with specific physical laws and that we should not separate the biological system from the physical one. More specifically, a comparative analysis has been done among 4 major plant divisions: Bryophyta, Pteridophyta, Gymnosperma and Angiosperma, and the classes of the animal phylum Chordata Vertebrata, the fish, the amphibia, reptiles, birds and mammals. The main concepts assumed for this study are the following. Our world is part of a quantum universe where the particle-wave duality, entanglement, non-locality, wave interference, superposition, and resonance of wave interactions allow extension of consciousness and mind to different spaces and time dimensions. The human brain is therefore assumed to act both as an interfacing organ and receiver that not only produces mind and consciousness, it can also be seen as an expression of a universal mind through holonomic communication with quantum fields. As such, phenomena like morphogenesis, convergence, and coevolution could well be explained without the use of dualism. The main differences between Darwinian evolution and quantum evolution now is that in Darwinian evolution mutations are random, whereas in quantum evolution quantum effects can bias the process of mutation towards providing an advantage for organism survival. That does not necessarily mean that they are exclusive, but most likely complementary. Now, there are a good number of examples of quantum evolution influence, such as the world of enzymes, immunity, antibiotic resistance, vision, olfaction, photosynthesis, respiration, bird navigation, most likely happening through teleportation or quantum tunneling. Now, we need to be familiar with the terms coevolution and convergence. So, coevolution refers to the evolution of at least 2 species, which occurs in a mutually dependent manner. The term coevolution is used to describe cases where 2 or more species reciprocally affect each other's evolution, and a good number of classic examples are shown below. Now, convergent and parallel evolution sometimes are confused. Convergent evolution is the independent evolution of similar features in species that are not closely related of different periods or epochs in time. Convergent evolution creates analogous structures that have similar form or function but were not present in the last common ancestor of those groups. So it's important to distinguish analogous from homologous structures, but also not to confuse with what we call parallel evolution, when 2 independent species evolve together at the same time in the same ecospace and acquire similar characteristics. But there are inconsistencies in convergent evolution, and there is lack of a standard set of techniques for quantifying its magnitude and testing its significance. Also here you can see an example of convergent evolution, that between the marsupials and the placentals. Now, possible mechanisms that exist behind convergence is most likely similar changes that happen at the genetic level, but the question is whether these genetic changes are preferentially fixed and why, and if it's due really to random mutation or not. There are, of course, studies that show that there's complex gene regulations in humans and mice, for example, where we have non-coding RNA sequences or jumping genes that regulate the process. Now, the methodology that I have employed in this study was a thorough literature review of theories of consciousness and evolution, in addition to choosing that hypothesis that consciousness indeed acts as a primary field of information underlying deep reality of space-time, and it dictates the evolution of all universal processes, including morphogenesis and the natural selection process of evolution, convergence and coevolution, utilizing both quantum and classical mechanisms of action, including backward causation. Specific adaptations of the aforementioned groups were compared and then recorded in a table followed by some analysis. So, to the best of my knowledge, the present perspective and comparison adds a new dimension which takes into account the possible role of consciousness being a unified and primary field of information, and thus guiding this evolutionary process in both at the classical level and at the quantum. So, I have studied a good number of structures that and adaptations among unrelated groups, and they are shown in red in the table that I'm going to present right now. The table is long, and someone needs a little bit of time to see it all, but I'm just going to point out that we have I've placed the geological age on the left, and then we have here the plant major divisions, and then the Chordata Vertebrata classes, and what I will go down to is these common adaptations and features that I am talking about. So mammals dominate today like the eudicote flowering plants do. Both groups share similar adaptations, like protecting their embryo in a seed enclosed in fruit, or by allowing full development of the embryo within the uterus, as the placenta mammals do. When it comes to birds, the birds share a lot of characteristics with the reptiles and have come up with hard-shelled egg instead of only leathery amniotic eggs. They also have similarities with the monocote flowering plants. Moving on to reptiles and gymnosperms, further back in time, those were the first to escape dependency on life in water. The adaptations of reptiles include tough, scaly skin, internal fertilization, the ability to conserve more water in their kidneys, and the development of amniotic eggs. Similarly, the gymnosperms developed cones and naked seeds which protected their embryo from desiccation, plus covered their epidermis with waxy material, allowing both groups to live solely on land. Further back in time, for both plants and animals, the evolutionary shift to land occurred at the end of the Devonian period. The key ancestor of the land-dwelling chordates at that time is the lobe-finned fish, and of course there is high dependence on presence of water for the divisions of the Bryophyta and Pteridophyta, that means the mosses and the ferns, as well as the amphibian class. Now, further back in time, in the age of fish, we have fertilization both in primitive plants and all fish subclasses, totally depending on the presence of water. So again, this table is available for anybody to check and study a little bit more. So the current study serves as a good example, I believe, of quantum evolution, convergence, and coevolution, as it focuses on similar adaptations and structures on those four divisions of plants and the classes of the Chordata Vertebrata. I have examined the role of course, gene drift, genetic recombination, epigenetic modifications, and genome duplication. It is definitely a preliminary study that can serve, though, as the basis for future research on morphogenesis, quantum evolution, and coevolution. So I propose certain, let's say, mechanisms here operating in the background of differentiation and morphogenesis, because it is a fact that morphogenesis is a problem of biological order, of unprecedented precision timing of correlated events of both spatial and temporal nature. And the real problem is this non-locality issue, how local interactions can control global development. So my question is, so is it the non-local morphogenetic field like that proposed by Rupert Sheldrake, the type of field that tells the particular DNA of a cell which part of the form-making plan to activate? And another question is, could these morphic fields be similar to those proposed by the quantum field theory or Bohm's implicate order? In conclusion, I have presented all of these things in relation to convergence and coevolution and made the comparisons. It is this present study is a new dimension that takes into account the possible roles of consciousness being a unified and primary field of information. For sure, all of these additional factors need to be examined, and this good number of structures and adaptations that I have examined and described and analyzed, their similarity in which could be very well explained if the hypothesis stated is indeed supported. So this case study has been used as a good example in support of the aforementioned hypothesis. The results of the comparison seem to support the hypothesis in a surprising yet obvious way. So this study may serve as yet another example suggesting evolutionary convergence across large phylogenetic distances where ancestral genomic repertoires render certain evolutionary trajectories more likely than others. So I finish by proposing certain suggestions and future recommendations in relation to either Darwinian evolution or quantum evolution. For sure, we have to take into consideration express sequence tags. We have to examine molecular transcripts and single nucleotide polymorphism and analyze those, cis elements, alternative splicing and promoter utilization, and antisense RNA, in addition to pseudogene activation, genome duplications, epigenetic modifications, genetic recombination, adaptive amino acid convergence, etc. For sure, this would lead us perhaps to a deeper level where interconnectedness allows the unfolding of forms and structures depending on the interactions with the environment and previous patterns or memories established. With this, I am finished, and I wish the best of success to all the participants of the conference and wish us all a brighter future. Thank you for your attention.
Hello, I'm Michael Beal. I'm presenting a paper on panspermia and consciousness. I'm currently a psychiatrist at the VA Medical Center in Cincinnati and on the faculty at the University of Cincinnati. Panspermia, meaning seeds everywhere, is the theory that microbes in space transmit life to habitable bodies and is required for consciousness to arise on Earth since its formation 4.5 billion years ago. The conservative materialistic paradigm requires the evolution of an adequately sophisticated nervous system to house an emergent consciousness. It's believed that Earth was not suitable for abiogenesis, the development of life from inorganic matter, until 3.5 billion years ago. This simply is not enough time to develop our complex biochemistry, therefore life on Earth was seeded from elsewhere. So let's talk about panspermia in some more detail. Basically, organic matter from space seeds the Earth, providing basic building blocks of life. Tons of space dust land on the Earth each day. This can be via light pressure, radiopanspermia, or via meteorites, lithopanspermia. This can be accidental or directed. Directed means that other life forms send life to us. However, what about other things like high-energy cosmic rays in the cold of space? Well, it appears viral structure is stable enough to survive, freeze-dried bacteria reconstitute, and there are organisms like Deinococcus radiodurans, which is a radiation-resistant bacterium. It has multiple copies of DNA, it has extra DNA repair enzymes, and is a model of how organic molecules may resist cosmic ray damage. Another example of a resistant animal that can survive outer space is the tardigrade, about 0.5 millimeter long. It can survive vacuum, extreme pressure, severe ionizing radiation, can be completely dehydrated and then rehydrate, and is able to forage and reproduce. Now, let's look at 3 arguments for panspermia and consciousness. First, even 4.5 billion years is not enough time to develop the complex biology of organic molecules, cellular structure, and cellular aggregation into animals, which is required for consciousness development. Complex biological structures must have been in process before the Earth existed. Second, there are multiple lines of evidence indicating biological materials are present in outer space and have been seeding the Earth. And third, directed panspermia is a theory that intelligent entities have directed the distribution of biological material. This theory presupposes that conscious entities intentionally distribute biological material throughout the cosmos to support life on other worlds. This emergent life on new worlds then develops a nervous system capable of supporting consciousness. According to abiogenesis, life comes from inanimate matter. Aristotle thought that life arose spontaneously and still arises spontaneously from inanimate matter through a process of abiogenesis. In the 1920s, Oprin and Haldane elaborated a theory of a primordial soup. In the 1950s, Urey and Miller elaborated on this by passing electric currents through inorganic chemicals and produced organic molecules, but these were in undifferentiated tars rather than in discrete organic molecules. Frozen conditions might create an RNA world. The steps to actual life forms, however, remains undefined. In 1973, Francis Crick wrote a paper on directed panspermia. He believed that beings from elsewhere had sent organisms deliberately to Earth. This solved the problem of very rare metals determining the structure of proteins and enzymes that otherwise were implausible on Earth. In their 1986 Nature paper on cosmic dust, Hoyle and Rick Ramasinghe found that the refractive index and emission band of polysaccharides best matched what was seen in space dust. And of course, polysaccharides form the cell wall of bacteria. Here we see a gaseous jet from a comet that crosses the Earth's orbit, an entirely plausible source of organic molecules from space. The famous Halley's Comet has an orbital period of 76 years. In its pass near the Earth in 1910, cyanogen gas was associated with it, and every May and October, meteors associated with its cometary orbit enter the Earth's atmosphere. In 2017, Oumuamua, an extrasolar comet, was noted to be passing through the solar system. It also was noted to exhibit non-gravitational acceleration as it accelerated away from the Sun. This is thought to be caused by so-called off-gassing, and of course off-gassing can leave extrasolar biological material behind. The Cambrian Explosion, which occurred 541 million years ago, may be clear evidence of panspermia. This was characterized by the sudden appearance of new complex animals. They had unique and variable body plans, and no precursor organisms could be identified. Elaborate genes in primitive organisms is suggestive of panspermia. Genetic developments like all genes being necessary for lungs being in place before lungs is an example. Genes that long precede their phenotypic expression is an example. Ancient bacteria have genes that are considered hallmarks of more recent cells. Loci DNA contains genetic instructions for actins, which form a skeleton-like framework in modern cells. Panspermia can make use of horizontal gene transfer, which is the movement of genetic material between existing unicellular and/or multicellular organisms. It's the primary mechanism of antibiotic resistance in bacteria, and in yeast, horizontal gene transfer is so common and frequent that it obscures vertical gene transfer. Are epidemics pathogens from space? The ancients believed that comets are vile stars and harbingers of doom, disease, death, and infection. Chinese astronomers recorded comets preceding plagues. The death of the dinosaurs 65 million years ago after an asteroid strike followed a pattern suggestive of epidemic disease. Smallpox is episodic, with epidemics widely separated in time. Is panspermia an aspect of the 1918 influenza epidemic? With widely different virulence and patterns of distribution, and simultaneous outbreaks in widely separated parts of the world, with onset the same day in Boston and Bombay. Complex life, as distributed by panspermia, requires more than the 13.5 billion years permitted by the Big Bang. Let's look at an aspect of that. In 1920, in the Great Debate, Harlow Shapley of Harvard argued that the Milky Way and its associated nebula constituted the entire universe. He lost the debate, but the question of the nature of the universe remained. Hubble, in 1929, proved that the Andromeda Galaxy was more than 1 million light-years distant. With this result, this was enough evidence to show that the Milky Way was just part of a much larger cosmos and not the entire cosmos itself. And a bigger universe implies a longer time. The Big Bang, the current theory of the universe, was created in 1925 following from the idea of Le Métrier, a Catholic priest who believed that the Milky Way had come from a primeval atom. And again, at this time, the Milky Way was the universe. The Big Bang is a peculiar hypothetical construction. It's a religious creation myth, but without conventional religious content. There's no apparent deity or a related religious theory of consciousness. And interestingly, the Big Bang label came from Hoyle, who was making fun of the idea, but oddly the Big Bang moniker stuck. The theory of a Big Bang universe requires the sudden expansion explosion from a singularity 13.5 billion years ago. It contains the conceit that the mathematical formalism of the Big Bang tells us exactly what was happening in infinitesimal fractions of a second after the Big Bang. It assumes that a redshift can only be a recessional velocity. The Big Bang insists on a strictly isotropic universe when all Big Bang theorists live on a big lumpy planet which is non-isotropic. The theory ignores that lumpy stars and galaxies are non-isotropic. And the Big Bang requires that 95% of the universe is dark matter and dark energy that we can't detect. It's quite clearly a creation myth of materialist scientism. The complexity of panspermia needs an eternal universe, or at least one that's been around for a really, really long time. Among the theories of an eternal universe are the steady-state universe of Hoyle, and Hoyle was the best astronomer of the 20th century. According to this, the universe must look the same to all observers at all places and times. Adding to this, Bondi and Gold hypothesized a new phenomenon, which included the spontaneous and continuous creation of matter within this steady-state universe. Electric universe theory relies on plasma, electrical currents, and magnetic fields, which work to concentrate matter and energy, making the universe the complex, dynamic, and uneven place that it actually is. Neo-Darwinism is a kind of biological probability. Darwin's original writings included concepts like inheritance of acquired characteristics as a means of change by evolution. By the 1940s, with no clear mechanism for genes to change from experience, Neo-Darwinism was born. The concept of random mutation of genes became the driving force for evolution. Randomness and probability became the control mechanism for genetic change and survival by natural selection. All gene sequences and the proteins they encoded were driven by chance alone. Walter L. Sasser is a physicist who turned to biology and introduced the concept of the immense number to biology. He's best known for the dynamo theory of the Earth's magnetic field. Late in his career, he attempted to apply the numerical principles of physics to the heterogeneity of biology, realizing the large number of states in biological systems. He defined the immense number, a dimensionless quantity, by taking the number of protons in the universe, as calculated by Eddington in 1938, 10 to the 80th power, and multiplying this times the number of picoseconds since the Big Bang, 10 to the 30th power, arriving at a number of 10 to the 110th power. He maintained that any systems with more states than this could not be calculated or characterized in detail. The proteome is a term for all of the proteins in an organism. The combinatorics of the proteome illustrate why we need panspermia for trillions of years to create our complex biology. The human proteome consists of between 20,000 and 6.2 million proteins, depending on focus. The lower number identifies those proteins produced from discrete protein encoding genes in human DNA. On average, these proteins are composed of 375 amino acids, with 21 amino acids used in their structure. That means that each protein has 21 to the 375th power, or 10 to the 495th power possible structures. The possible variations in the proteome then are 10 to the 495th power to the 20,000th power, which gives us a number of 10 with an exponent consisting of nearly 54,000 zeros. This slide has 2,500 zeros. That means we would need nearly 21 slides of this sort to illustrate all of the zeros in the hyper-immense number of the proteome exponent. The 110 numbers of Elsasser's just 10 to the 110th power exponent are illustrated on the top in red. The number of possible states of the human proteome is a hyperimmense number and is a partial and incomplete description of the complex and combinatoric scale of biology. Linear strings of amino acids must fold in order to make functional proteins, with the estimate of 10 to the 26 possibilities for the folding of each protein structure. All proteins exist in the context of coding by DNA using specific cellular structures which are even more complex. And this just scratches the surface for the complexity of just a single cell. So, has the cosmos been around for trillions of years until consciousness evolved as epiphenomenal of life, or has consciousness been there all along? So, if life required trillions of years of development, in advance of life, were there other complex or subtle energy forms or complex structures capable of consciousness? With immense age needed in the universe for life to evolve, are consciousness and cosmos simply coexistent? Is the consciousness that we take for granted as biological entities a more limited and prosaic state compared to what exists in the cosmos as a whole? From this point of view, panspermia can be seen as a conceptual tool to indicate that consciousness must have a fundamental place in the universe. Thank you.